This video is to complement male reproductive function notes starting on page 213 of your notebook. We've already covered the male structures, so what we're really dealing with here is the formation of sperm cells and then a little bit to deal with testosterone production. So right here, just starting with our cover page, we have a review of some structure. We have a testis, so this is the male gonad. And then within it, we see a couple different things. This outer layer is the tunica albuginea. There's a connective tissue. It penetrates into the testis. And so these are septa. The areas that they're coordinating off are lobules, and each lobule has 1 to 4 seminiferous tubules. So in here we have a series of really tightly coiled tubes. And these tubes are where the sperm are going to be produced. They lead to an area in sort of the hilus of the testicle called the rete testis. That leads to a storage area called the epididymis. We're going to see that again. And then from there, once the sperm have matured, they can be released into the vas deferens. So the process of making sperm is called spermatogenesis. So literally sperm creation. This begins at puberty, and millions are produced every day within the seminiferous tubules. This is a difference from female reproduction, where a woman, when she's born, her ovaries already contain the follicles with the immature eggs. Now those eggs don't begin to mature until puberty, but the eggs are already present. For males, the sperm aren't there present. They don't start being produced until puberty. And then millions are produced every day. So now, what are we looking at? This is one of those seminiferous tubules cut in half, and we're looking down the center of it. So here in the middle, it's labeled lumen. This is the hollow space. This is the thickness of the tissues, the cell layers that make up the tube itself, and this is the outside of the tube. So in here, we see a few things labeled for us. One of them is spermatogonium, and these are stem cells that are going to eventually become the sperm. We also see some other types of cells that are listed here, which is maybe more important at the college level, but something we're going to not really deal with today. So spermatogonia are primitive stem cells. What are stem cells? Generally speaking, they are cells that still have the ability to become different types of cells. They haven't differentiated yet. They haven't completed sort of a developmental pathway. So they're still kind of flexible in what they're able to become. These are located in the outer edge of the seminiferous tubule. So let's back up for a second and just look at that slide again. They're in the outer edge of the tubule, and they're going to work their way inward into this lumen. So before puberty, these stem cells go through rapid mitotic division. So basically, they clone themselves. They go through prophase, metaphase, anaphase, telophase. And, they're starting with 46 pairs of chromosomes, and they're ending with 46 chromosomes, 23 pair of chromosomes, ending with 23 pair. Before puberty, follicle-stimulating hormone gets secreted in increasing amounts. Now remember that in the endocrine system, we talked about follicle-stimulating hormone and luteinizing hormone being produced by the anterior pituitary. We're going to see that again in a minute. Follicle-stimulating hormone in a woman is going to literally do what it says, stimulate the follicles. We're going to see that in the female reproductive unit. For males, we'll kind of come back to that. But this is being secreted in increasing amounts. So spermatogonia can produce 2 types of cells. So when a cell produces or clones itself and makes new cells, we sometimes call those daughter cells. So the original was the stem cell, the spermatogonia, but then it can make a daughter cell. So there's type A and type B. The type A stem cell remains within that tubule, and it's always there to keep making more of itself. So really, it's a population that clones itself and always stays as stem cells. Now, what's the other type do? Well, that other type doesn't stay in that region. It starts to move, and as it gets pushed towards the center of the tubule, it begins to specialize and it begins to go through the stages of meiosis and become what's called a primary spermatocyte. So, again, here is a testis. These are in green, the seminiferous tubules. They're zooming in on it. It's a cross section. So we're looking down it. So here's the hollow space. Okay. Here's that thickness of the tubing. And they've labeled some of these things for us. Spermatogonia. So there are the stem cells and they're working their way in. Here are some early spermatids. So early sperm. Notice that they're closer to this center hollow area than the other cells, which are at the outer edge. Now looking at the stages up here, here's a stem cell, a spermatogonium. It has 2N. What does that mean? N is our species number for how many chromosomes we should have. So for humans, that would be 23. And then it's 2N because we have 23 pair of those chromosomes. So if you were going to do mitosis, you're going to start with one cell. End result is you have two cells, and they are identical in terms of their chromosome number to the original. So here what we're doing is mitosis. This is cloning itself and cloning itself and cloning itself. That is those type A daughter cells. I am maintaining a constant population of spermatogonium or stem cells. Now, some of those can go on and become sperm. So the ones that go on to become sperm are now referred to as a primary spermatocyte. So it has twice the number it should need, or the full number that a normal cell would have. When it goes through meiosis, it does that reduction division. So now I've got our secondary spermatocytes, and that is a cell that has half the normal number. So these are now haploid. These cells up here are diploid. And you may be familiar with those terms. So what this means is here's one whole set of chromosomes. It completes its division. And you should remember in meiosis, we start with one cell. We end with 1, 2, 3, 4 cells. We start with the full number of chromosomes and we cut that in half. So each of these is now going to be able to go on and become a functional sperm eventually. So at this point, we call them spermatids. They don't necessarily look like sperm yet. That's a different step. So these each have one whole set of chromosomes. So you're going to get one set of chromosomes from mom, one set from dad, put them together, and that makes the fertilized egg. And that would mean one N comes from a sperm, one N comes from an egg, put them together, you get the original 2N back, but in a different combination. So now it's got to go through another step. We did spermatogenesis here. Now we have to go through something called spermiogenesis, which will make them actually be what you would think of as a sperm. So again, this is happening in the seminiferous tubules, and we've seen this before. So the primary spermatocyte is the stem cell that undergoes meiosis to form 4 sperm. What is meiosis? A special type of nuclear division. So nuclear division meaning in the nucleus of the cell. What's in the nucleus? That's your DNA. That's your chromosomes. So meiosis is something that only really occurs in the gonads. So whenever we talk about meiosis, we're really talking about making sperm or eggs. So in this case, we're talking about making sperm. So what's the end result? 4 gametes, 4 sex cells, 4 spermatids. So here we are. This is, again, inside a seminiferous tubule. Here is a stem cell. That stem cell moves on to become a primary spermatocyte. It moves on to become a secondary spermatocyte. Those become spermatids. and those are going to then be released into this hollow space here to be transported. So what's a spermatid? It is the sex cell that was produced by spermatogenesis. Some facts about it: contains half the number of chromosomes. So for a human, that means that sperm has 23 chromosomes. What's it going to do? Unite with an ova to form a fertilized egg, which is going to be called a zygote. And we'll cover that in another day. So now, once it's fertilized, you have the full 46 chromosomes that you should have to do proper development. Where are these spermatids located? In the lumen of the tubule, in the hollow space of the seminiferous tubules. What do we know about them, though? They're non-functional. They're non-modal. So what's modal mean? Modal means movement. So they can't swim by themselves yet. So they're going to get propelled down the seminiferous tubules from the epididymis. It's even once they're able to be functional sperm, they're not actually activated until they get the right chemical signal. So mostly they get moved within the body of the male through peristalsis. But we do have our spermatids. We want to really finish their completion of their development. So this is called spermiogenesis. What do? They're going to get rid of extra cytoplasm because they want to become streamlined, and they're going to develop 3 areas: a head, a midpiece, and a tail. So here's the stem cell. It's got 2 sets of chromosomes here as a primary spermatocyte. Goes down, now it has half the normal number. Those split and they become the spermatid. So I have 4 spermatids, each with 23 chromosomes. But they don't look like a sperm. I see a little bit of flagella, but there's a lot of cytoplasm in here. So what they then do is shape themselves to have a head, a midpiece, and a tail. So the head is where the DNA is located. Now the end of it is a cap called an acrosome. So it's anterior to the nucleus of the head, meaning it's the very tip of the sperm. And it's kind of like a large lysosome, meaning it has digestive enzymes in it. And the function of that is when they come in contact with the follicle cells that surround and protect that egg that's been ovulated, they are what penetrate through it. We need to break down those follicle cells for the sperm to get in. The midpiece has centrioles, which in the cell are normally used for cell division in moving those spindle fibers. So in this case, they can cause some movement for us. And what they're moving are the filaments, in other words, the tail. And this is also where there's some mitochondria located. The mitochondria is important. This is where we do our respiration. So it is taking in a source of glucose, for example, or fructose, and it's using it to produce ATP. That cellular energy then is powering the tail to swim. So we've got a head. Inside the head is the nucleus. In here, there are 23 chromosomes. There's an acrosome. That's going to be a digestive enzyme that can break down the follicles. In the midpiece, there are the centrioles and the mitochondria, and then we've got the tail. So spermatogenesis form primary spermatocytes and then release them immaturely into the lumen. And this takes 64 to 72 days, but they can't swim. So they're moved by peristalsis, where they get stored in the epididymis, and that's where they mature. So then from there, it would then receive the signal and then be released through the body, through the vas deferens to the urethra. Kind of just backing up for 1 second, we will talk about this when we do fertilization, but there's mitochondria in this section, but this section doesn't enter the egg. Only the nuclear contents enter the egg. So even though the sperm does have mitochondria, the mitochondria doesn't get into the egg. So you do get all of your mitochondria from your mother. Now again, function-wise, our male gonads have an endocrine and an exocrine function. So we just talked about the exocrine function, the production of sperm. Now the endocrine function is making the androgens or testosterone. So this is done by those interstitial cells, the major hormones that we get stimulated from the anterior pituitary, which we can see located right here in the region of the sella turcica, are FSH and LH, follicle stimulating hormone and luteinizing hormone. So these names are more appropriate descriptors when we see female anatomy, but males do have these same hormones. So here is the diencephalon of the brain. The bottom of that, in there we have the hypothalamus. Hanging from that, we've got the pituitary. So follicle stimulating hormone in women is going to stimulate the maturity of a follicle to release with an egg that could then go on to complete development and potentially be fertilized. So here, there's no follicles to stimulate, but that's what stimulates the sperm. So they do have a similar function in that they both stimulate the gonads to produce the gametes. Luteinizing hormone targets those interstitial cells and causes it to produce testosterone, but we often don't call it luteinizing hormone in the male. We call it interstitial cell stimulating hormone, ICSH. Same thing, different name. And then what does testosterone do once it's made? It goes on and then triggers secondary sex characteristics. So remember, from the endocrine system, there are three ways to trigger a hormone. You have hormonal, meaning hormones go on to a target and cause that thing to release other hormones. So hormones trigger hormones. We had humoral, which is really like concentrations of things in your body fluid. So blood sugar would be an example, salt levels. And then neural. Obviously, our nervous system can stimulate certain glands to produce things like our adrenal glands. So in this case, um, the luteinizing hormone triggers production of testosterone. Testosterone goes on and triggers secondary sex characteristics. Okay, so here in our hypothalamus, we have the anterior pituitary, follicle stimulating hormone, targets cells that deal with the seminiferous tubules and deal with sperm production. Luteinizing hormone or um, interstitial cell stimulating hormone goes on to target and make testosterone. What are the secondary sex characteristics for males? Deepening voice, mostly due to an enlargement, a physical enlargement of the larynx. Increased hair growth in the body, especially axillary, pubic, and facial regions. Um, Enlarged skeletal muscle, increased density of the skeleton, increased thickness of bones. And some of these changes can go on into the early 20s even. So just because a male has hit puberty in his early teens, possibly before, it could be into age 22, 23, 24 before some of these changes have completed themselves, especially in terms of muscle development and skeleton development. And that completes this coverage of this section of the notes. If you have any questions, let me know.